Okay, in this lecture, we are going to see life across 18 orders of magnitude. One thing we want to stress, this lecture is not about memorizing scales. It's about recognizing where different kinds of modeling becomes possible. So when you are modeling a drug binding, you are working at nanometer scale. When you are thinking about drug distribution in tissue, you are at what millimeter scale, different scales, different physics, different modeling approaches. So we will walk through all 18 orders of magnitude so you know where your model works. So let's start with electron. You won't model electrons directly in drug discovery, but their behavior underlies everything above the scale. A bond formation, partial charges, hydrogen bonding, and molecular orbitals all arise from electron distributions. So only specialized quantum methods like DFT or QMMM simulate them explicitly when accuracy really matters for predicting reactions. But most ML models for drug discovery simply ignore this scale. Now, at 10 to the power of minus 15 meter scale, that's proton, nucleons, the same thing. You don't model at this scale. The main relevance is maybe isotopes, deuterium labeling for mass spec experiments, radioactive tracers for imaging. This layer simply sets the physical constants that chemistry and everything above it runs on. Now let's go to atom. So this is where chemistry starts. Atomic properties, electronegativity, size, valence electrons determine bonding. In practice, this is the lowest level layer most drug discovery models touch. Molecular fingerprints, RD-kit features, and graph neural networks. All of these start by representing a molecule as a graph of atoms with the properties attached. If you are modeling small molecules, this is your basic unit of information. Now let's talk about the small molecules. They are typically 50 to, 10 to 50 atoms, generally under 500 Daltons. So these are your building blocks. Amino acids polymerize into proteins, then nucleotides polymerize into DNA, RNA, and then fatty acids form membranes. When you are doing a small molecule drug design, you are working at this scale. Your model predicts how a molecule at this scale interacts with a protein that is 10 times bigger. Now let's talk about macromolecules. Proteins, nucleic acids, and lipids, they sit at nanometer scale. Proteins are usually 5 to 10 nanometer across, hundreds to thousands of amino acids folded into precise 3D structures. When you are modeling protein drug interactions, or protein interactions, or maybe antibody design, RNA, so this is your scale. Methods like AlphaFold, for example, operate at this layer, map sequence to 3D structure. Now let's check next layer, molecular machines. So the molecular machines like ribosomes, proteosomes, they sit at tens of nanometers scale. The ribosome is about 20 nanometers, made of dozens of proteins plus RNA. The proteasome is another large complex that degrades damaged or unwanted proteins. Antibiotics like tetracycline, for example, target the bacterial ribosome. Cancer drugs like proteasome inhibitors, for example, and they target proteasome. Modeling these requires understanding multi-component assembly and dynamics. Traditional docking approaches work here, but you are dealing with larger, more flexible systems than single proteins. Now, next layer, viruses. Viruses span a wide range of sizes from around 20 nanometers up to nearly a micron, or even more actually. I think there is a virus that is almost 4 micrometer. That's crazy. So AAV, adeno-associated virus, shown here is about 25 nanometers. It's used for gene therapy because it can package a DNA and deliver it to cells. What we need to think here is viruses are essentially packaged genetic information plus a delivery mechanism. When you are designing AAV vectors for gene therapy, you are engineering at this scale. And then you are optimizing the capsid proteins for specific tissue targeting. And then next scale, we will discuss bacteria. Bacteria, they are about a micron, roughly 1,000 times bigger than a small molecule, and then maybe around 100 times bigger than a typical protein. So they are completely self-contained systems. When you are developing antibiotics, you need to kill bacteria without harming human cells. That's a specificity problem at the cellular level. Some ML models try to predict antibiotic activity, but it's harder than protein drug binding because you are pretty much modeling a whole system response. And then now we will see eukaryotic cells. So let's say human cells are about 10 to 30 microns. They are 10 times bigger than bacteria and much more complex. There is membrane-bound nucleus, specialized organelles, internal trafficking systems. When you are designing small molecule drugs, you need to get them into these cells to the right compartment. A drug targeting a nuclear protein needs to cross the cell membrane and then the nuclear membrane. Your molecule might bind great in vitro but fail because it cannot get to where it needs to be. When you are designing cell therapies, you are engineering entire cells. You can go through bunch of organelles on this slide. And then next, let's talk about tissues. So tissues are at millimeter to centimeter scale. This is where you start thinking about drug distribution. After dosing, a drug has to move through real physical structures, blood vessels, extracellular space, and dense tissues. We can take an example in a solid tumors. The outer layer gets exposed, but the core, often doesn't, simply because vasculature is poor. So this is why PKPD, pharmacokinetics and pharmacodynamics modeling matters. A compound can be perfect on binding and potency, but it still, let's say, if it never reaches the target tissue at an effective concentration, it fails in vivo, like you can guess. Now, let's talk about organ systems. At this scale, you are dealing with the whole organism. When you give a drug, it goes everywhere. The liver metabolizes it, the kidneys filter it out, the heart pumps it around. The immune system might respond to it if it looks foreign. Drug interactions happen here. One drug changes how another one is metabolized. This is why clinical trials fail. A lot of times you are dealing with a whole body system with feedback loops and off-target effects. Now let's see full stack organism. A human is about a meter tall, 37, let's call 40 trillion cells, trillions of bacteria in the microbiome, constant immune surveillance, regeneration, repair, all these things happening simultaneously. You cannot model all of this. That's why drug discovery uses staged models. You would test binding in vitro, and then you would test cell killing in the cell culture, and then maybe efficacy in animals, then small human trials, and then larger trials. Each stage adds complexity. Your computational models usually work at one or two layers of the stack. Not all 18 orders of magnitude at once. It's just not possible. Next, we will just see all scales together. Look at these numbers. You have what, some 3 times 10 to the power of 13 cells, about 22 macromolecules, and then 10 to the power of 28 electrons. So the complexity explodes as you go down in scale. This is why bottom-up simulation of a whole human from first principles is just not possible. Instead, we build models at the right level of abstraction. Molecular dynamics simulates atoms and forces. That's good for nanosecond timescales and nanometer spaces. Cellular models abstract away molecular details. Organ models abstract away cellular details. You pick the right level for what you are trying to predict. And then now briefly we will see some modeling examples and then some scales. Different scales need different modeling approaches because the dominant physics changes. At atomic scale, quantum mechanics and bonding matters. At protein scale, structure and conformational dynamics matter. At cellular scale, you are doing systems biology, so, like gene networks, signaling pathways, those kinds of things. At tissue scale, you are dealing with the PKPD, distribution and clearance. At organism scale, you are doing clinical modeling with the population statistics. As you design therapeutics, you need to think about which scales matter for your particular problem. Thank you.